Yes. So in the last class, we completed up to conditions for growth. Conditions for growth. Am I right? Yes. So from plant growth and development beginning to up to conditions of growth, do you have any questions? Do you have any doubts? So in the last class, we teach from introductory part to conditions. In that, we discussed so many concepts. What is meant by growth? Is growth is determinate or indeterminate? Is growth is measurable or not? What are the different phases of growth? What is meant by growth rate? How many types of growth rates are present? What is arithmetic growth? What is geometric growth? And what are the conditions of growth? These are the concepts that we discussed in the last class, students. So somebody is asking, growth is determinate or indeterminate? Yes, growth is indeterminate in plants. That is the main axis of the plant body. But some organs in the plant, such as leaves, flowers, buds, and fruits, show determinate growth because they drop off frequently, periodically, and new flowers, new fruits, new buds will come. So growth may be determinate or indeterminate, but the main axis that what we are talking about is indeterminate, and some organs are determinate. I think clear, right? So generally growth is indeterminate, no doubt about that. And some are asking about what is AGR, what is RGR. Yes. You have to take what is the growth per 1 day. So in 1 week, some 14 centimeters are grown, for example. So what is the growth actually? 2 centimeters per day. That's why you have to take. And somebody asking, what is exponential growth? So lag phase is exponential growth. Log phase is exponential phase. The phase where the organism grow rapidly is the exponential growth, also called that log phase. And somebody is asking again, what is meant by AGR? What is meant by RGR? I will tell you briefly again before we are going to discuss about the today's content. Okay. AGR means absolute growth rate. RGR means relative growth rate. Okay. Measurement and comparison of growth occurs actually in unit time is called as absolute growth rate. actual growth per unit time is absolute growth rate. Actual growth occur per unit time is called absolute growth rate. Measurement and comparison of growth per unit time is called absolute growth rate. Okay. For example, a plant grow to 14 centimeters in a week. So its absolute growth rate per day is 2 centimeters. That is absolute growth rate. Final growth minus initial growth. And what is RGR? Relative growth rate. Absolute growth rate on initial basis is called relative growth rate. Absolute growth rate on initial basis is called relative growth rate. So, when comparing with initial growth, how much growth occurs? This is on initial basis, if you want to say, that becomes relative growth rate. I hope you understand. Relative growth rate. So, absolute growth rate and relative growth rate. Measurement and comparison of growth on initial basis is called relative growth rate. Therefore, relative growth rate is equal to absolute growth rate by initial size into 100. Initial size into 100. This is what is called relative growth rate. Yes. Students, I think you understood regarding RGR and AGR, because some students are asking what is RGR and what is AGR. Clear now? And someone is asking that growth occurs due to cell division. Yes, 100%. Growth is caused due to cell division in the meristematic regions. So there are two types of growth: linear growth and growth in girth, called secondary growth. Growth in length is called linear growth. Growth in girth is called secondary growth. Okay, clear? That's all. And now we will continue the next topic, that is differentiation, de-differentiation and redifferentiation. This is very, very important as far as the NEET examination is concerned. So let us discuss these three phenomena. One is differentiation. Second one, de-differentiation. And redifferentiation. Redifferentiation. Differentiation, de-differentiation and redifferentiation. Very, very important, and they show some confusion, but please carefully follow. All of you know there are several meristems like root apical meristems present at the tip of the root, shoot apical meristem present at the tip of the stem and branches, and cambium. cambium. That may be vascular cambium or corku-cambium. So there are meristems in the plant body. Ram and Sam are apical meristems. They are responsible for linear growth of the plant. That is, growth in length. Linear growth, growth in length. Growth in length. And there are lateral meristems like vascular cambium, corku-cambium are lateral meristems. They are responsible for secondary growth. Because in any plant, generally in dicots and gymnosperms, not in all plants, secondary growth is present. But primary growth is present in all plants. Right? So primary growth is caused by primary meristems called Ram and Sam, and secondary growth is caused by lateral meristems called vascular cambium and corku-cambium. Okay. That means the property of a meristem is to divide and form new cells. The property of any meristem is always divide and form new cells. Okay. Therefore, from these meristems, several cells are derived. Several cells are derived from apical meristems, from lateral meristems. New cells are continuously being formed. These cells, not all cells, some cells, differentiate and become mature. Differentiate and mature. This act of maturation from meristematic cell is called as differentiation. Differentiate and becomes mature. This act leading to maturation is called as differentiation. This act leading to maturation, this act leading to mature is called differentiation. So, several cells are formed from these meristems. These meristems undergo changes and gradually transformed into mature cells. This formation of this leading to maturation of the cells is called differentiation. Understand, students? During this process, the cells undergo several changes. The changes include changes in the cell wall, changes in the protoplasm. So major changes takes place. Changes occurs in the cell wall, changes occurs in the protoplasm. Protoplasm changes, cell wall changes. For example, one meristematic cell changes into tracheary elements. Tracheary element. One meristematic cell now changes into tracheary element, tracheary cell. That is a mature cell. This is a young cell. This is a living cell. This cell changes into tracheary element. For that, this cell undergo several changes. First change is they loses protoplasm. They loses protoplasm. As a result, the cell becomes dead cell. A cell without protoplasm is a dead cell. Understand? A dead cell. First change, protoplasm is lost from the cells. Now, the cell walls becomes thick. The cell walls gradually thickened. Lignin depositions takes place and transformed into a rigid wall. A rigid wall. Cell wall becomes thick, inelastic, and the cell wall become hard due to the deposition of lignin. Lignified walls are formed because in the tracheary elements, tracheids, water is transported, water and minerals are conducted from root system to shoot system. Therefore, the walls must be strong. That is why cell wall becomes thickened. Cell wall becomes hard. And for that, deposition of lignin takes place. Cell wall becomes thick. No protoplasm. And finally, one cell that's spherical shaped, more or less isodiametric cell is a meristematic cell. This isodiametric cell, a meristematic cell, changed into an elongated spindle-like, tube-like cell with thick lignified walls and no protoplasm in the cell. So this living cell become a non-living cell, a mature cell, a dead cell. The shape changes. Living cell become dead cell. Here the cell walls are thin cellulosic. Here thick lignified walls are formed. And this is a soft and this is a very hard rigid cell. And this can resist during the movement of water from root system to shoot system. Understand? This act of maturation from meristematic cell is called as differentiation. Just I told you one example, only one example. What are the changes that takes place during the conversion of a meristematic cell into a tracheid cell or a tracheary element? Meristematic cells are living cells, tracheids are dead cells. That means in order to convert that living cell into dead cell, it must undergo several changes. And finally, this leads to what is called as differentiation. Students, I hope you understand. No protoplasm means no cell organelles, no nucleus, just cell wall is present. And in that there is a space. This space is called as lumen. That is called a lumen, through which water is transported. Understand? Okay, this is a living cell. They contain protoplasm. They contain thin walls. But this cell is transformed from meristematic cell. It is a non-living cell. It is a dead cell. It won't contain protoplasm. The cell is elongated, but here is isodiametric. This is elongated. and the cell wall is thick and lignified. And these changes leads to formation of this cell. Now this cell is formed by differentiation process. Like that, many cells in the body of a plant, for example, are formed by differentiation process. How a vessel is formed from a meristematic cell. They undergo certain changes. And epidermal cells are formed by differentiation. Cortical cells are formed by differentiation. Understand? So, depending upon that type of cell and the location of the cell, the meristematic cell want to become which type of cell, accordingly they undergo changes. And this is what is called as differentiation, students. Hope you understand what is meant by differentiation. Most of the cells in the plant body are formed by differentiation. Permanent parenchyma cells are formed by differentiation. Very important point. Parenchyma is a permanent tissue. All parenchyma cells are formed from meristematic cells by differentiation. Collenchyma is formed from meristematic because once upon a time all these are meristematic cells. The meristematic cells undergo differentiation. It may become a parenchyma, or it may become a collenchyma, or may become a fiber, or may become a scleroid, or may become a xylem, or may become a phloem. Like that, because embryo contains meristems. So meristems are present in the embryo. This embryo undergo differentiation. During differentiation, several changes occur. Ultimately, that meristematic cell changed into some other cell, depending upon their position, their location. That cell is formed. Students, do you have doubt? Any doubt regarding differentiation? So most of the cells in the plant is formed by differentiation. Yes, primary xylem. Yes, we are talking about primary xylem. Primary xylem, primary phloem is formed from meristematic cells of the embryo by differentiation process. By differentiation process. That's all. That is about differentiation. Hope you understand, everyone. So embryo contain undifferentiated cells. Gradually, during the formation of the body, differentiation takes place. So most of the cells in the body are formed by differentiation process. Correct. Now we will discuss about de-differentiation. De-differentiation. What is meant by de-differentiation? Let us see. De-differentiation. What is meant by de-differentiation? Let us see. If differentiated cell again differentiated, if a differentiated cell again differentiated and form new cell, it is called de-differentiation. Already I told you that meristematic cell undergo differentiation and form a mature cell. This process is called differentiation. For example, meristematic cell form a trachid cell. So meristematic cell become mature cell. The mature cell do not divide further generally, but sometimes This differentiated cell again regain the activity of division. They again, that means they regain the activity. Already this is a differentiated cell. Generally they do not differentiate. Whenever the situation demands, it has to differentiate again. Then only the cell again differentiate and form another cell. That is meristematic cell and become meristematic. This is called de-differentiation. After differentiation, cells are formed. They form the plant body. They are the mature cells. They are restricted to perform a particular function. Generally, they won't divide further. But whenever the cell demands, some cells may undergo differentiation further and becomes a meristematic. The permanent cell, you can call it as this permanent cell. Mature cell is nothing but permanent cell. Meristematic cell become mature cell. The mature cell again differentiate and become meristematic cell. This is called de-differentiation. Students, I think you are following, right? Yes. Formation of a permanent cell from a meristematic cell is differentiation. Okay. Now this permanent cell do not divide usually further, but some cells regain the activity of cell division, and transformed into meristematic cell. This process is called de-differentiation. For example, between the vascular bundles in the dicot stem, see this? These are the vascular bundles in the dicot stem. Okay. This is one vascular bundle, another vascular bundle, another vascular bundle. These are the vascular bundles. Between the vascular bundles in dicot stem, there are parenchymatous cells called medullary rays. Parenchyma cells called medullary rays. These medullary rays are formed by differentiation process. Medullary rays are nothing but parenchyma. Okay, this parenchyma during secondary growth, they de-differentiated again. They de-differentiated and form a cambium between the vascular bundles like this. This cambium is called interfascicular cambium. So, interfascicular cambium is a meristem. Medullary rays are permanent cells. Understand? So I will write here. Meristematic cell undergo differentiation and form medullary rays. So this is a permanent tissue. This is a permanent cells. These are permanent cells. Now what I am telling is, this permanent cell dedifferentiated, and form interfascicular cambium. This is meristematic layer. Formed between the vascular bundles in the dicot stem only. So meristematic cell become permanent cell. Again the permanent cell regain its activity and become meristematic. The second time, divided, this is called as dedifferentiation. This is the best example of dedifferentiation in plants. The best example for the dedifferentiation is this one. Differentiation of medullary rays into interfascicular cambium is an example for dedifferentiation. Differentiation of medullary rays into interfascicular cambium. Differentiation of medullary rays into interfascicular cambium is called dedifferentiation. I am telling very slowly, students, because this will create a lot of confusion. So in order to get more clarity, I am telling several examples. So slowly I try to follow and understand. Okay? Yes. This is one example. I told you one example. One example is formation of interfascicular cambium, not intrafascicular cambium. Intrafascicular cambium directly formed from differentiation process. That is primary in origin. This is secondary in origin. Intrafascicular cambium, primary in origin. Directly embryonic cells become intrafascicular cambium. Here embryonic cells become medullary rays first. Some medullary rays, not all medullary rays. Some medullary rays become dedifferentiated and form one more cambium, meristematic layer called interfascicular cambium. This is one aspect. Next, this is the scalar part. You see now, in the dicot stem only, this region is called cortex. That is the cortex. So I told you secondary growth occurs in two phases. Secondary growth occurs inside the stele, stellar secondary growth. Outside the stele, extrastellar secondary growth. Outside the stele, extrastellar secondary growth. Inside the stele, intrastellar secondary growth. Understand? Secondary growth occurs inside the stele, stellar secondary growth. Outside the stele, extrastellar secondary growth. intrafascicular cambium is formed by differentiation. Yes, correct, 100%. So I will show you the previous slide also. After completion of this one, I will show you. Now, in the cortex, one more cambium is formed. In the cortex, one more cambium is formed like this. In the cortex, one more cambium is formed. This cambium is called cork cambium. This cambium is called cork cambium. And you know, this cortex is nothing but parenchymatous tissue. Parenchyma is a permanent tissue. And this is formed from differentiation of meristematic cells of the embryo. Differentiation of meristematic cells of the embryo. So, cortex is parenchymatous. Thus, cortex is formed by differentiation process. Now, some cells of the cortex de-differentiated and form cork cambium. So, cortex is a permanent cell, and that permanent cells again de-differentiated and form meristematic cells called cork cambium. For example, see, meristematic cell becomes meristematic cells become parenchyma cells. Meristematic cells become parenchyma cells. This process is called differentiation. Okay? And generally, parenchyma cells are permanent cells. They are restricted to perform a particular function. They are restricted to perform a particular function. But sometimes during secondary growth, some cells of the parenchyma becomes de-differentiated and form cork cambium. Differentiated and form cork cambium. So, formation of cork cambium from parenchyma cells is de-differentiation. Formation of interfascicular cambium from medullary rays is by de-differentiation. So, interfascicular cambium, cork cambium are examples of de-differentiation. Interfascicular cambium and cork cambium are examples of de-differentiation. Hope you understand. Students understand differentiation process. A differentiated cell again differentiated. This is called de-differentiation. A differentiated cell once again differentiated. This is called de-differentiation. What is meant by de-differentiation? Formation of a meristematic cell from a permanent cell is called de-differentiation. Formation of A meristematic cell from a permanent cell is called de-differentiation. You can say like that. Formation of a permanent cell from meristematic cell, differentiation. Formation of a meristematic cell from permanent cell, de-differentiation. Yes, differentiated cell, again differentiated. That is called de-differentiation. Yes, cortex is a parenchyma. Interfascicular cambium, cork cambium are formed due to de-differentiation. Exactly 100% correct. And that is very important neat bit also. Understand? Interfascicular cambium, cork cambium are formed due to de-differentiation. So examples of de-differentiation process: interfascicular cambium and cork cambium. Interfascicular cambium is formed between the vascular bundles in the stellar part, and cork cambium is formed in the cortex region. Cork cambium is formed in the cortex region. And one more point also I want to add here. For example, tumor formation. Whatever it may be, a tumor in an animal cell or a plant cell, because of attack of bacteria, tumors may be formed in plants. Tumor formation is an example for differentiation or de-differentiation or re-differentiation. A differentiated cell again differentiated and form a tumor. Right? So tumor formation is an example for de-differentiation. How can you say that formation of tumor takes place by de-differentiation process? Do you agree that tumor formation is a de-differentiation process, students? Do you agree that tumor formation is a de-differentiation process? Yes, you are absolutely right. Tumor cells are parenchyma cells. Parenchyma cells are formed by differentiation. Now again they are de-differentiated and form the tumor. Therefore, yes, very good. So I think now, yes, you don't have any confusion regarding differentiation and de-differentiation. No confusion regarding differentiation and de-differentiation. Okay, formation of parenchyma, formation of cholenchyma, formation of sclerenchyma, formation of vascular tissues, formation of epidermis, formation of cortex, all these are takes place by differentiation process. So only certain tissues undergo de-differentiation. And similarly, one more example. Formation of callus in tissue culture. Formation of callus in a tissue culture is an example for. Formation of a callus in tissue culture is an example for. Can you say? Yes, that is also example for dedifferentiation. In tissue culture, we will take an explant. We will take an explant. What is an explant? Any part of the plant is taken in tissue culture to grow into a new plant is called explant. Correct? Explant may be a root cell, explant may be a stem cell, explant may be a leaf cell, any part of the plant that is generally formed by differentiation process. This explant taken into the test tube having the medium, this is the explant already formed by differentiation process. They dedifferentiate and form a mass of undifferentiated mass of tissue called callus. We will take explant that is already a permanent cell, divide and form a callus, undifferentiated mass of tissue called callus. Okay. So this is what is called callus formation in tissue culture. Both are example for dedifferentiation process. So now we go with next one called redifferentiation. 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 Differentiation of dedifferentiated cell is called redifferentiation. Differentiation of a dedifferentiated cell is called redifferentiation. Understand? Differentiation of a dedifferentiated cell is called redifferentiation. Already that is formed by dedifferentiation process. Now again differentiated and form a permanent cell. This is called redifferentiation. So product of differentiation, permanent cells. Product of dedifferentiation, meristematic cells. Product of redifferentiation, permanent cells. Hope you are following. Hope you are following, right? Product of differentiation, permanent cells. Product of dedifferentiation, meristematic cells. Product of redifferentiation, permanent cells. I will show you the sequence. I will show you the sequence. Meristematic cell becomes permanent cell in continuation. I am talking permanent cell. This is called differentiation. Permanent cell again differentiated and form meristematic cell, and this again differentiated and form a permanent cell. A permanent cell. The first process is called differentiation. The second process is called dedifferentiation. Third one is called redifferentiation. Students, this is very, very important. Meristematic cell become permanent cell. Differentiation. Permanent cell become meristematic cell. De-differentiation. That meristematic cell formed by de-differentiation again undergo differentiation called as redifferentiation and form the permanent cell. Now I will explain redifferentiation process now. So differentiated cell again differentiated. For example, I am talking about redifferentiation. Redifferentiation. Meristematic cell formed by dedifferentiation become differentiated. Example 1. Interfascicular cambium. Interfascicular cambium formed in the secondary growth in dicot stem between the vascular bundles produces interfascicular cambium, combines with the fascicular cambium and form vascular cambium, and form the vascular cambium divides and form secondary xylem towards inner side, also called wood, and secondary phloem towards outer side, also called bast. This process is called de-differentiation. Formation of secondary xylem and secondary phloem are examples of de-differentiation. Students, please follow. See. Hope you understand, all of you. Formation of secondary xylem and secondary phloem, that is wood and bast, are examples of de-differentiation, redifferentiation. So interfascicular cambium formed by de-differentiation. These are the redifferentiated products. Sorry, these are the redifferentiated products. This is redifferentiation. Redifferentiation, yes. Next, one more example also I will tell you. Example 2. Cork cambium formed by de-differentiation undergo redifferentiation and form cork or phellum towards outer side and secondary cortex or felloderm towards inner side is also by redifferentiation. Students, I think you are following right now. So formation of cork and secondary cortex are example for redifferentiation, because cork cambium undergo redifferentiation and form cork or phellum, and secondary cortex or felloderm. Other names for that. Cork is also called phellum, secondary cortex is also called felloderm. So totally I told you 4 tissues. 4 tissues. Examples of redifferentiation: secondary xylem, secondary phloem, cork and secondary cortex. Examples of redifferentiation: wood, bast, phellum, phylloderm. These are the technical words. So these 4 are the only examples of redifferentiation in plants. Understand? So these are the examples of redifferentiation. These are the examples of dedifferentiation. Interfascicular cambium, cork cambium are formed by dedifferentiation. Secondary xylem, secondary phloem, cork and secondary cortex are formed by redifferentiation. I hope all of you understood regarding redifferentiation. Example of differentiation: parenchyma, collenchyma, sclerenchyma, etc. Example of dedifferentiation: intervascular cambium, cork cambium. Example of redifferentiation: secondary xylem, secondary phloem, cork and secondary cortex. Clear? Primary xylem and primary phloem are formed by differentiation process. Primary xylem, primary phloem are formed by differentiation process. No, differentiation process, that is cork cambium, vascular cambium, is formed in the plants that undergo only secondary growth. Understand? That means these dedifferentiation, redifferentiation is not seen in all the plants. Only in the plants which show secondary growth, that is dicots and gymnospermic plants. No, it occurs in the root also. But in the root, what is not directly, the vascular cambium is formed. Vascular cambium is directly formed. In the stem, first intravascular cambium, then intervascular cambium. Partly primary, partly secondary. But in case of root, directly vascular cambium is formed, cork cambium is formed. They are secondary in origin, secondary in function. Okay. Now, after this, what do you understand is, growth process in plants is open. So, growth in plants is open. I told you already. That means it may be indeterminate or determinate. Indeterminate or determinate. Growth in plants is open. I told you what is open form of growth in plants. If growth occurs due to continuous formation of new cells because of the presence of meristems, it is called as open form of growth. So generally the growth is indeterminate, but some organs of the plant are determinate. So open form of growth in plants is due to meristems. Absolutely. So growth is open in plants because of the presence of meristems. Now we can say that differentiation in plants, is also open. Differentiation in plants is also open. So growth is open. Differentiation is also open because the cells formed from the same meristem, from one meristem, several cells are formed. The meristem undergo cell division and form, several cells are formed. The cells formed from same meristem form different structures at maturity. Different structures at maturity. That is why differentiation is also open. Try to understand this word. Why differentiation is also open? Assertion, differentiation in plants is also open. Assertion is true. Reason. The cells formed from the same meristem. The cells formed from the same meristem differentiate and form different structures at maturity. This is also correct explanation of A. Growth is open in plants, correct. Differentiation is also open in plants because cells from the same meristem form different structures at maturity. Cells from the same meristem form different structures at maturity. Therefore, the growth is also, the differentiation is also open. Moreover, this differentiation depends upon location of the tissues formed. The function of that tissue depends upon its location. The functions of that cells or the tissues depends upon its location. For example, the meristematic cells present towards outer side form root cap.